See, basically your cells are constantly working, no? They need oxygen to break down food molecules like glucose, amino acids, fatty acids. This gives energy for everything you do, running, thinking, even sleeping. But when cells use oxygen, they produce carbon dioxide. Now, carbon dioxide is harmful for the body. So, oxygen must be continuously provided and carbon dioxide must be continuously removed. This exchange of O2 from the atmosphere with CO2 from cells is what we call breathing, or commonly we say respiration. So, this whole process taking in oxygen from air and releasing carbon dioxide, we give it a name, breathing. Some people call it respiration. Same thing. And understand this, it is not something you do once and forget. It is continuous. Every second, every millisecond, your cells need oxygen and your body produces CO2. So breathing must happen continuously. This is absolutely essential for living. Now, different animals have different ways of getting oxygen depending on where they live and how complex they are. Let me start with simple ones. Sponges, coelenteroids, flatworms, these are very basic animals, no? They are so simple that oxygen and carbon dioxide just diffuse directly through their entire body surface. No special structures needed, just diffusion. And earthworms, they use their moist skin, their cuticle. Because their skin is always wet, gases can diffuse through it. Now insects, they are more advanced. They have this network of tiny tubes inside their body called tracheal tubes. These tubes carry air directly from the atmosphere into the body. Very clever system, no? Then you have aquatic animals. Fish, crabs, they live in water. How can they get oxygen from air? They cannot. So they use gills. Gills are these special vascularized structures that extract oxygen dissolved in water. That is branchial respiration. The word branchial means gills. Now, when we come to land animals, especially the more complex vertebrates, they use lungs. Lungs are vascularized bags, meaning they have blood vessels supplying them. Amphibians like frogs, reptiles like snakes, birds, and mammals like us, we all use lungs. Of course, fishes use gills because they live in water. But here is something interesting. Amphibians like frogs can use two methods. They have lungs, but they can also breathe through their moist skin. We call this cutaneous respiration. Cutaneous means skin. Okay, so when you breathe in, where does the air go? First, it enters through the nostrils. You have a pair of nostrils, two of them opening out above your upper lip. These lead to the nasal passage and then to the nasal chamber. Simple? No. This is where your breathing journey starts. And important thing, the air gets cleaned and warmed right here in the nasal chamber. Some foreign particles get trapped. From the nasal chamber, air goes to the pharynx. Now the pharynx is very interesting. It is a junction point. It is the common passage for both food and air. Meaning what? When you eat, food goes through pharynx. When you breathe, air goes through pharynx. Then the pharynx opens into the larynx region and from there into the trachea. Now the larynx, it is a cartilaginous box. Cartilaginous means it is made of cartilage. This is your sound box. This is what produces your voice when air passes through. Now here is something very important. When you swallow food, it should go down to the esophagus, not into your lungs, no? How does this happen? There is a small flap called epiglottis. Think of it like a small door. When you swallow, this epiglottis closes and covers the glottis, the opening to the larynx. This prevents food from entering the larynx and going to the lungs. Very clever mechanism. If epiglottis did not work properly, you would choke. Food would go into windpipe. So this thin elastic cartilaginous flap is absolutely essential for safety. So air comes down from the larynx into the trachea. The trachea is a straight tube like a pipe, no? It extends down to the middle of your chest cavity. We call it mid-thoracic cavity. Now, at a specific point at the level of the fifth thoracic vertebra, this trachea divides. It splits into two branches, the right primary bronchus and the left primary bronchus. One goes to right lung, one goes to left lung. Now, each bronchus does not just stop there. It divides again and again and again. This is what we mean by repeated divisions. From each primary bronchus, we get secondary bronchi. From secondary, we get tertiary bronchi. And then from tertiary, we get bronchioles. These keep dividing until they become very, very thin. almost microscopic. These finest tubes are called terminal bronchioles, like tree branches, no? Main trunk divides into big branches, then smaller branches, then twigs. Now you might wonder, these tubes are very thin. Will they collapse when we breathe? No, because they are supported by cartilaginous rings. Think of it like the rings inside a vacuum cleaner hose, no? These incomplete cartilaginous rings prevent the tubes from collapsing. The trachea has them, the bronchi have them, and the initial bronchioles also have them. But as we move deeper into the lungs toward the alveoli, these rings become less frequent because the tubes become wider and more flexible. From each terminal bronchiole, something very important happens. Multiple tiny structures form. These are called alveoli. Alveoli are like tiny little sacs, bag-like structures. Very important thing to understand. They have very thin walls, extremely thin. And they are not round and perfect, they are irregular shaped. But here is the crucial part. They are highly vascularized. This means blood vessels surround them. This is where actual gas exchange happens. Oxygen diffuses through the thin wall into the blood. Carbon dioxide diffuses from blood into the alveoli. So all of this, the bronchi, the bronchioles, the alveoli, they all together form the lungs. The lungs are like a branching network, very complex. And you have two of them, right lung and left lung. They are inside your thoracic cavity in your chest, and they are the main organs for respiration. Now, the lungs are not directly touching the chest wall. Why? Because they are covered by something called pleura. Pleura is a double-layered membrane, like two sheets, and between these two sheets, there is pleural fluid. You know what this does? When your lungs move during breathing, they rub against the chest wall. If there was no fluid, there would be friction, pain. But the pleural fluid acts like a lubricant, reduces friction. Very smooth movement. The outer pleural membrane touches the thoracic lining, gyrate. That means the inner wall of the chest cavity. The inner pleural membrane directly touches the lung surface. Now let me teach you a very important concept. The respiratory system has two parts. We call them conducting part and exchange part. The conducting part includes everything from the nostrils all the way down to the terminal bronchioles. So the nostrils, nasal chamber, pharynx, larynx, trachea, all the bronchi, all the bronchioles, all of this is conducting part. What does it do? It conducts air, no? But there is more to it. As air moves through, foreign particles get trapped. The air gets humidified, means moisture is added, the air gets warmed to body temperature. But the actual exchange of gases, the real purpose of respiration, happens in the exchange part. The exchange part is only the alveoli and the ducts connected to them. This is where the magic happens. Oxygen comes from the air in the alveoli and goes into the blood. Carbon dioxide comes from the blood and goes into the air in the alveoli. This is the actual gas exchange. And why is it so efficient here? Because the alveolar walls are extremely thin. This thin barrier between air and blood allows rapid diffusion. Okay, think about the structure of your chest cavity. It is called thoracic chamber and it is absolutely airtight. Very important this. Now what forms this airtight chamber? On the back, we say dorsally mihi is the vertebral column, your spine. On the front ventrally is the sternum. You can feel it in the middle of your chest. On the sides laterally are the ribs and on the bottom is the diaphragm, a muscular sheet shaped like a dome. Now here is something very clever about this design. Because the thoracic chamber is airtight, whenever the thoracic volume changes, the lung volume changes too. Simple. If thoracic volume increases, lung volume automatically increases. If thoracic volume decreases, lung volume automatically decreases. You understand why this is so important? We cannot directly pull the lungs or push the lungs, no? We can only change the thoracic cavity size. And because they are connected in this airtight system, lung volume changes automatically. This is the principle on which breathing works. Now, respiration, the complete process, involves 5 steps. Let me go through them one by one. The first step is what we commonly call breathing, or more technically, pulmonary ventilation. In this step, atmospheric air containing oxygen is drawn into the lungs through the nose or mouth. At the same time, air rich in carbon dioxide is pushed out from the alveoli. This is purely mechanical, means it involves muscles and volume changes. This is what happens every time you breathe in and breathe out. Step 2 happens right inside the lungs. It is the diffusion of gases across the alveolar membrane. The oxygen from the air in the alveoli diffuses through the thin membrane into the blood. At the same time, carbon dioxide from the blood diffuses into the alveoli where it can be breathed out. This diffusion happens because of pressure gradient. Oxygen pressure is higher in alveoli, carbon dioxide pressure is higher in blood. Step 3. Transport of gases by blood. Now the oxygenated blood needs to reach all the tissues of the body. The oxygen is carried primarily by a protein called hemoglobin in the red blood cells. Similarly, carbon dioxide is carried by blood. We will learn later the different ways carbon dioxide is carried. But the point is, the blood acts as a transport medium. It carries oxygen from lungs to all tissues and carries carbon dioxide from tissues back to lungs. Step 4. Diffusion at tissues. When oxygenated blood reaches the tissues, oxygen diffuses from blood into the tissue cells. Why? Because oxygen pressure is higher in blood than in tissue. At the same time, carbon dioxide which has accumulated in the tissues diffuses into the blood. Why? Because carbon dioxide pressure is higher in tissues. Again, it is pressure gradient doing the work. Step 5 and final step, cellular respiration. The oxygen reaches the cell and the cell uses it. It uses oxygen to break down food molecules, glucose, amino acids, fatty acids, and extract energy. This energy powers all cellular activities. But in this process, carbon dioxide is produced as a harmful byproduct. This carbon dioxide needs to go out. And that is why the whole cycle starts again. That is why we keep breathing. Each step connects to the next step. Beautiful process, no? So breathing has two parts. First is inspiration, also called inhalation. This is when you breathe in. Atmospheric air is drawn into the lungs. Second is expiration, also called exhalation. This is when you breathe out. The air in the alveoli, which is now rich in carbon dioxide, is released out. Both of these happen because of pressure differences, pressure gradient between the lungs and the atmosphere. Now understand this basic principle. Air moves from higher pressure to lower pressure. Always think about it. When you open a balloon and let air out, the air rushes out because pressure inside balloon is higher than outside. Same principle here. For inspiration to happen, the pressure inside lungs must become less than the atmospheric pressure. Then air rushes in from outside. For expiration to happen, the pressure inside lungs must become greater than atmospheric pressure. Then air rushes out. So how do we create these pressure differences? By changing volume. More volume means less pressure. Less volume means more pressure. Okay, let us understand how inspiration happens. It is initiated by contraction of the diaphragm. You remember the diaphragm? It is at the bottom of the thoracic chamber, dome-shaped. When it contracts, it flattens and moves downward. This movement increases the thoracic volume in what we call the antero-posterior axis. Antero-posterior means from front to back. So the front to back distance of the thoracic cavity increases. Think of it like a container airway. If you pull the bottom down, the container gets bigger vertically. But that is not all. At the same time, other muscles called external intercostal muscles contract. Where are they? Between the ribs. Intercostal literally means between the ribs. When these muscles contract, they pull the ribs upward and also lift the sternum. When ribs go up and out, the side-to-side diameter of the thoracic cavity increases. This is in the dorsoventral axis. Dorsoventral means from back to front or in terms of distance, top to bottom orientation. So now you have two things happening. Vertical space increases due to diaphragm and side-to-side space increases due to external intercostal muscles. So the overall effect. The total volume of the thoracic cavity increases significantly. And remember what we learned earlier? Because the thoracic cavity is airtight. When thoracic volume increases, lung volume automatically increases too. Now increased volume means decreased pressure on food. It is a law of physics. So the intrapulmonary pressure, the pressure inside the lungs becomes less than the atmospheric pressure outside. And what happens then? Air rushes in from high pressure outside to low pressure inside the lungs. Air flows in, that is inspiration. Now let us understand expiration. It happens when the inspiration muscles relax. The diaphragm relaxes and moves back upward to its resting position. The external intercostal muscles relax, so the ribs go back down, and there are internal intercostal muscles. They can help push the ribs further down. So both the diaphragm and ribs return to their original positions. This is basically the reverse of what happened during inspiration. During normal quiet breathing, this is a passive process, means the muscles are not actively contracting, they are just relaxing. When the muscles relax and return to rest position, what happens? The thoracic volume decreases and lung volume also decreases. They are connected. Remember, decreased volume causes increased pressure. So the intrapulmonary pressure increases and becomes greater than the atmospheric pressure. Now air pressure inside is higher than outside. Air flows out, it is pushed out from the lungs. This is expiration. Now during normal quiet breathing, expiration is passive, no? But when you need to breathe out more forcefully during exercise, during deep breathing, you can use additional muscles. There are muscles in the abdomen. When they contract, they push the contents of the abdomen upward. This pushes the diaphragm even further upward. This increases the strength of expiration. More air can be expelled. This is forced expiration, and it uses additional muscles beyond the basic intercostal muscles. On average, a healthy person breathes about 12 to 16 times per minute. That is the normal resting rate. But this is not fixed. During exercise, you breathe faster, maybe 30, 40 times per minute. When you are relaxed or sleeping, it might slow down. You can also voluntarily control your breathing. You can take slower, deeper breaths or faster, shallower breaths. But ultimately, the breathing rate is regulated by your brain, specifically by respiratory centers that we will learn about later. Now, how do doctors know if your lungs are functioning properly? They use an instrument called spirometer. Spirometer measures the volume of air you breathe during different types of breathing. You breathe into it and it records how much air comes in and goes out. It can measure your tidal volume, your reserve volumes, your capacity. We will learn these terms next. By looking at these measurements, doctors can assess if your lungs are functioning normally or if there is some problem. If someone has asthma or emphysema or any lung disease, the spirometry results will be abnormal. So it is a very important diagnostic tool. Let me teach you about respiratory volumes. First is tidal volume. Tidal volume is simply the volume of air you inspire or expire during one normal quiet breathing cycle. Not forced breathing, just normal relaxed breathing. For a healthy person, this is approximately 500 milliliters. Now if you breathe 12 to 16 times per minute and each breath is 500 milliliters, you can calculate that is about 6,000 to 8,000 milliliters of air per minute. Significant volume, no? Next is inspiratory reserve volume or IRV. This is the additional volume of air that you can forcefully breathe in after you have already done a normal inspiration. Means what? After you normally inhale, if you try extra hard to suck in more air, how much extra can you get? That is IRV. It is like a reserve. For a healthy person, this is approximately 2,500 to 3,000 milliliters. Quite a lot, no? So your lungs can hold much more than just the normal tidal volume. Similarly, there is expiratory reserve volume or ERV. This is the extra volume of air you can forcefully breathe out after you have already done normal expiration. After you normally exhale, if you try extra hard to push out more air, how much can you push? That is ERV. For a healthy person, this is approximately 1,000 to 1,100 milliliters. Lesser than IRV because during expiration, the lungs cannot completely collapse. There is always some air left. Now, very important residual volume or RV. This is the volume of air that remains in your lungs even after the most forceful expiration. No matter how hard you try to push air out, you cannot push out this volume. Why? Because the airways are not completely collapsed. Some air always remains trapped inside the alveoli. For a healthy person, this is approximately 1,100 to 1,200 milliliters. And this is actually good. If this residual volume was not there, the alveoli would collapse like deflated balloons and they would stick together. The lungs would become non-functional. So residual volume is essential for maintaining lung inflation and structure. Now we can combine different respiratory volumes to get what we call pulmonary capacities. These are more useful measurements than individual volumes because they give us a better picture of lung function. and they are very important for doctors to diagnose if there is any lung disease. Inspiratory capacity, or IC. What is the maximum amount of air you can breathe in starting from your normal resting position? You can breathe in your normal tidal volume, and you can also breathe in extra using your reserve. So IC equals tidal volume plus inspiratory reserve volume. That is PV plus IRV. This comes to approximately 3500 milliliters. This is the total air you can inspire. Expiratory capacity, or EC. Similarly, what is the maximum air you can breathe out starting from normal inspiration? You breathe out your normal tidal volume plus your expiratory reserve. So EC equals TV plus ERV, approximately 1500 milliliters. Not as much as inspiratory capacity because you cannot completely empty the lungs due to residual volume. Functional residual capacity, or FRC. After you normally exhale, how much air remains in your lungs? It includes the expiratory reserve volume plus the residual volume. So FRC equals ERV plus RV. This is approximately 2200 to 2300 milliliters. This is the amount of air that is always present in your lungs at rest. This air is important because it provides a buffer on the seal and dilutes incoming air, and it prevents oxygen concentration from dropping too much between breaths. Vital capacity, or VC. This is very important. Vital capacity is the maximum volume of air a person can breathe in after a forced expiration. Or you can think of it the other way, the maximum volume after forced inspiration. In other words, this is all the air that can be exchanged. It includes expiratory reserve volume plus tidal volume plus inspiratory reserve volume. ERV plus TV plus IRV. This comes to approximately 4800 milliliters. And this is clinically very important because it indicates if the lungs are functioning normally or if there is disease. Finally, total lung capacity, or TLC. This is all the air your lungs can hold at maximum. It includes residual volume, expiratory reserve volume, tidal volume, and inspiratory reserve volume. RV plus ERV plus TV plus IRV. Or more simply, vital capacity plus residual volume. This comes to approximately 6000 milliliters for a healthy adult. This is the complete lung volume, everything your lungs can hold. If this is reduced in a patient, it suggests their lungs are somehow restricted or damaged. Now, let us talk about where gas exchange happens. The primary site is the alveoli. Remember, alveoli are those tiny air sacs at the end of the bronchioles. These are the sites where oxygen from the air enters the blood and carbon dioxide from the blood enters the air. Why are alveoli so perfect for this? Because they have very thin walls, almost transparent, and around them are blood capillaries. So you have air on one side and blood on the other side. Separated by just a thin barrier, gases can easily diffuse across, but gas exchange also occurs between blood and tissues. When oxygenated blood reaches tissues, oxygen diffuses from blood into the tissue cells where it is needed. At the same time, carbon dioxide, which has accumulated in tissues, diffuses into the blood to be transported back to lungs. This happens everywhere in the body, in your brain, heart, muscles, skin, everywhere. So there are two main sites of gas exchange, one in the lungs and one in the tissues. How does this exchange happen? By simple diffusion. This is a passive process, no energy is needed. Molecules simply move from where there is more of them to where there is less of them. Either we talk about concentration gradient or pressure gradient may rise. Both mean the same thing. A gas will diffuse from area of high concentration to area of low concentration, or from high pressure to low pressure. Now, the rate of diffusion depends on several factors. First is the solubility of the gas. More soluble gases diffuse faster. Second is the thickness of the membrane. Thinner membrane means faster diffusion. That is why alveolar walls are so thin to maximize diffusion. Third is the surface area. Larger surface area means faster diffusion. Lungs have millions of alveoli, giving huge surface area. Fourth is the concentration or pressure gradient. Steep gradient means faster diffusion. All these factors are arranged in your body to maximize gas exchange. Alright, now let me explain partial pressure. When you have a mixture of gases like atmospheric air, which has nitrogen, oxygen, carbon dioxide, and other gases, each gas contributes its own pressure to the total. The pressure from oxygen is called partial pressure of oxygen. The pressure from carbon dioxide is called partial pressure of carbon dioxide. and so on. So partial pressure is simply the pressure contributed by one individual gas in a mixture. We use special notation for partial pressures. For oxygen, we write PO2 NES, that is P followed by O2. For carbon dioxide, we write PCO2 NES, that is P followed by CO2. The P stands for partial pressure. These values are expressed in units like millimeters of mercury or kilopascals. And very important, NES, these partial pressure values are different in different places in the body. In lungs, they are different. In blood, they are different. In tissues, they are different. These differences are what drive gas movement. This is key concept. Let me give you the actual numbers. In atmospheric air, the air around us, the partial pressure of oxygen is 159 millimeters of mercury. The partial pressure of carbon dioxide is 0.3 millimeters of mercury. Very low carbon dioxide, right? Because carbon dioxide is just a trace gas in the atmosphere. This is the air we breathe in from outside. Now in the alveoli inside the lungs, the partial pressure of oxygen is 104 millimeters of mercury. This is lower than in the atmosphere because in the alveoli, oxygen has already started to move into the blood. Also, oxygen mixes with the residual volume urea and the air that never left the lungs. The partial pressure of carbon dioxide in alveoli is 40 millimeters of mercury, much higher than in atmosphere because carbon dioxide has diffused in from the blood. This difference between atmospheric and alveolar pressures is important to know. For victile on lazi sanitary. Now in the alveoli inside the lungs, the partial pressure of oxygen is 104 millimeters of mercury. This is lower than in the atmosphere because in the alveoli, oxygen has already started to move into the blood. Also, oxygen mixes with the residual volume urea, the air that never left the lungs. The partial pressure of carbon dioxide in alveoli is 40 millimeters of mercury, much higher than in atmosphere because carbon dioxide has diffused in from the blood. This difference between atmospheric and alveolar pressures is important. It allows oxygen to continue entering blood in the lungs. In deoxygenated blood coming back from tissues that has given up its oxygen, the partial pressure of oxygen is 40 millimeters of mercury. Very low, right? Because the oxygen has been used by the tissues. The partial pressure of carbon dioxide is 45 millimeters of mercury, higher than in alveoli. So there is a pressure gradient. Mercury and oxygen pressure is higher in alveoli than in deoxygenated blood, so oxygen goes into blood. Carbon dioxide pressure is higher in blood than in alveoli, so carbon dioxide comes out of blood. After the blood picks up oxygen in the lungs, it becomes oxygenated blood. In this oxygenated blood, the partial pressure of oxygen is 95 millimeters of mercury, increased from 40, and carbon dioxide partial pressure is now 40 millimeters of mercury, same as in alveoli, because carbon dioxide has left the blood in the lungs. This oxygenated blood then travels to tissues. Finally, in tissue cells, the partial pressure of oxygen is 40 millimeters of mercury, same as in deoxygenated blood. Why? Because tissues are actively using oxygen. The oxygen is being consumed faster than it arrives, so pressure stays low. The carbon dioxide pressure is 45 millimeters of mercury, higher than in oxygenated blood, which has 95 millimeters of oxygen, partial pressure, and 40 millimeters carbon dioxide. So oxygen flows from blood into tissues, carbon dioxide flows from tissues into blood. Beautiful gradient system, no? Now look at the oxygen gradient. In atmospheric air, it is 159. In alveoli, it drops to 104. In oxygenated blood, it is 95. In tissues, it is 40. See the pattern? It keeps decreasing. Oxygen always moves from higher pressure to lower pressure, from alveoli to blood, from blood to tissues. This is progressive decrease. And at the tissue level, oxygen pressure drops to 40 below. Very low, because cells are consuming it. This low pressure pulls more oxygen out of blood. Carbon dioxide is opposite. In tissues, it is 45. In venous blood, it is also 45. In alveoli, it drops to 40. In atmosphere, it is barely 0.3. So carbon dioxide flows from high pressure in tissues to lower pressure in lungs. At each step, pressure decreases slightly. This drives carbon dioxide out through the lungs and into the atmosphere. So we have beautiful countercurrent system. Oxygen goes in one direction, carbon dioxide in opposite direction. Both systems work together. Here is a very important point. Carbon dioxide is much more soluble in blood than oxygen. How much more? 20 to 25 times more. This means that for every unit of pressure difference, much more carbon dioxide will diffuse compared to oxygen. So even though the partial pressure changes for carbon dioxide are smaller than for oxygen, carbon dioxide actually diffuses faster and more efficiently. Nature has arranged this so carbon dioxide, which is a metabolic waste, is efficiently removed. Very smart system. Now let me tell you about the barrier between air and blood. It is called the diffusion membrane and it has 3 layers. First layer is the thin squamous epithelium of the alveolus. This is the wall of the alveolus. Second layer is the endothelium of the alveolar capillary. This is the wall of the blood vessel surrounding the alveolus. Third layer is in between. It is called basement substance. This is made up of basement membranes that support both the alveolar wall and the capillary wall. So you have air side layer, blood side layer and a supporting layer in between. Now how thick is this membrane? It is much less than 1 millimeter. It is incredibly thin. Think about it, the distance for oxygen to travel from air to blood is less than 1 millimeter. Carbon dioxide from blood to air is also less than 1 millimeter. This thinness is absolutely essential. It means gas molecules have a very short distance to diffuse. They cross quickly and efficiently. In fact, I can say that all factors in your body are against to favor diffusion of oxygen from alveoli to tissues and carbon dioxide from tissues to alveoli. The membrane is thin. Check. The alveoli have huge total surface area, millions of them. Check. The walls are thin. Check. There are constant blood capillaries supplying oxygen and removing carbon dioxide. Check. Pressure gradients are maintained. Check. Everything is optimized for gas exchange. This is why respiration is so efficient in our body. Now, blood is the medium that transports gases. But how much of each gas is transported by what method? Let us start with oxygen. About 97% of oxygen is transported by red blood cells. More specifically, it binds to a protein called hemoglobin and is carried as oxyhemoglobin. Only 3% of oxygen is carried in a dissolved state in the plasma, so almost all oxygen rides on hemoglobin 50 and the most Now, blood is the medium that transports gases, but how much of each gas is transported by what method? Let us start with oxygen. About 97% of oxygen is transported by red blood cells. More specifically, it binds to a protein called hemoglobin and is carried as oxyhemoglobin. Only 3% of oxygen is carried in a dissolved state in the plasma, so almost all oxygen rides on hemoglobin 50. 97%. Very efficient. Carbon dioxide is different. It is transported by 3 different methods. 20 to 25% binds to hemoglobin as carboaminohemoglobin means hemoglobin carrying carbon dioxide. 70%, the majority is carried as bicarbonate ions, HCO3-. Very clever system because bicarbonate is water soluble and can be efficiently transported in plasma. And 7% is dissolved in plasma. So carbon dioxide uses multiple pathways. Why? Because it needs to be removed efficiently from tissues and transported to lungs for exhalation. Hemoglobin is a very important protein. It is red in color. That is why blood is red. It contains iron atoms and it is present in huge quantities in red blood cells. Each red blood cell contains about 270 million hemoglobin molecules. These hemoglobin molecules have the special ability to bind reversibly with oxygen. Reversibly means what? They can pick up oxygen and later release it, not permanent binding. This reversible binding is perfect for oxygen transport. When oxygen binds to hemoglobin, we call the complex oxyhemoglobin. Oxy means oxygen. Hemoglobin means the protein. So oxyhemoglobin is oxygen-carrying hemoglobin. One hemoglobin molecule can carry a maximum of 4 oxygen molecules. Why 4? Because hemoglobin has 4 iron atoms. Each iron atom can bind with 1 oxygen molecule. So theoretical maximum is 4 oxygen molecules per hemoglobin. Though in practice, it varies depending on conditions. Now, whether oxygen binds to hemoglobin or not depends on several factors. The primary factor is the partial pressure of oxygen. Where oxygen pressure is high in lungs, oxygen readily binds to hemoglobin, forming oxyhemoglobin. Where oxygen pressure is low in tissues, oxygen releases from hemoglobin. But there are other factors that modulate this binding. Higher carbon dioxide pressure favors oxygen release. Higher hydrogen ion concentration means low pH, also favors oxygen release. Higher temperature also favors release. These factors work together. In the lungs, where conditions are favorable, oxygen binds. In tissues where conditions are different, oxygen is released. Very intelligent system. When scientists studied how oxygen saturation of hemoglobin varies with partial pressure of oxygen, they got a very interesting curve. It is S-shaped. We call it sigmoid shape. Sigmoid means S-like. So it is the oxygen dissociation curve. On one axis, we plot the percentage saturation of hemoglobin. NES means what percentage of the 4 oxygen sites are occupied. On the other axis, we plot the partial pressure of oxygen. The resulting curve has an S shape. What does this curve tell us? At low oxygen pressure, saturation is low oxygen does not bind much. As pressure increases, saturation increases. At high oxygen pressure, like in the lungs, saturation is very high, almost all 4 sites occupied. The middle steep part is interesting. Small change in pressure causes large change in saturation. This is the physiological range where tiny changes in conditions cause oxygen to load or unload. This curve is extremely useful. By looking at different oxygen dissociation curves drawn at different carbon dioxide pressures, different pH values, different temperatures, we can understand how these factors affect oxygen binding and release. The curve shifts right when carbon dioxide is high or temperature is high or pH is low, venting, meaning oxygen is more easily released. The curve shifts left when these conditions reverse, venting, meaning oxygen is held more tightly. This is the basis for understanding respiration adjustments in different body conditions. In the alveoli of the lungs, conditions are ideal for oxygen to bind to hemoglobin. Why? Because there is high oxygen partial pressure, 104. There is low carbon dioxide pressure, which means high pH. The blood is slightly alkaline and temperature is lower in the lungs. All of these conditions work together to promote oxygen binding to hemoglobin. Oxygen readily picks up, forms oxyhemoglobin. But in the tissues where cells are actively metabolizing, conditions are opposite. There is low oxygen pressure because cells are using it down to 40 millimeters of mercury. There is high carbon dioxide pressure because cells are producing it up to 45. There is high hydrogen ion concentration. The pH is lower, slightly acidic, and temperature is slightly higher in active tissues. All these conditions work together to push oxygen off from hemoglobin. Oxygen releases from oxyhemoglobin. So here is the beauty of the system. Oxygen picks up where it is abundant in the lungs. It travels in blood attached to hemoglobin as oxyhemoglobin. When it reaches tissues where oxygen is scarce and carbon dioxide is abundant, it automatically releases. Every 100 milliliters of oxygenated blood delivers approximately 5 milliliters of oxygen to tissues under normal conditions. The system is self-regulating based on local conditions. No external control needed. One way carbon dioxide is transported is by binding directly to hemoglobin. When carbon dioxide binds to hemoglobin, we call it carbaminohemoglobin. Note this is different from oxyhemoglobin. Carbon dioxide does not bind at the same sites where oxygen binds. It binds at different sites on the hemoglobin protein. About 20 to 25% of the carbon dioxide transported by blood is in the form of carbaminohemoglobin. So hemoglobin has 2 important jobs. It carries oxygen and it also carries carbon dioxide. A multifunctional protein.
In the tissues where carbon dioxide pressure is high and oxygen pressure is low, carbon dioxide readily binds to hemoglobin. This is where carbon dioxide loading occurs. The high carbon dioxide pressure drives the binding. The low oxygen pressure is also important. It means oxygen is being released at the same time, leaving space for carbon dioxide to bind. The Haldane effect, the effect of oxygen saturation on carbon dioxide carrying capacity.
But when blood reaches the lungs, carbon dioxide conditions change. Carbon dioxide pressure is now low at 40. Oxygen pressure is very high, 104. These conditions cause carbon dioxide to dissociate from hemoglobin. The carbon dioxide leaves and oxygen takes its place. This is dissociation. The released carbon dioxide diffuses into the alveoli where it can be breathed out. Beautiful cycle in tissues, carbon dioxide loads in lungs, it unloads. Now the most important carbon dioxide transport system involves an enzyme called carbonic anhydrase. This enzyme is present in very high concentrations inside red blood cells. There are also minute quantities in the blood plasma. What does this enzyme do? It catalyzes, speeds up the reaction between carbon dioxide and water. Without this enzyme, the reaction would be too slow for efficient transport. With this enzyme, it happens almost instantaneously. The reaction is CO2 plus H2O reversibly converts to H2CO3 glut, that is carbonic acid glut, which reversibly converts to HCO3 minus glut is bicarbonate ion plus H plus with hydrogen ion. The two arrows show this is reversible, can go forward and backward. This reversibility is crucial for both loading and unloading carbon dioxide. In the tissues, there is lots of carbon dioxide because cells are respiring, using oxygen and making carbon dioxide. This carbon dioxide diffuses into red blood cells. Once inside, the enzyme carbonic anhydrase converts carbon dioxide to bicarbonate. The bicarbonate is charged HCO3 minus. It leaves the red blood cell and enters the plasma. This is how most carbon dioxide is transported as bicarbonate in plasma. About 70%, and there is an interesting exchange showing the chloride ions enter the red blood cell when bicarbonate leaves. We call this chloride shift. Now when blood reaches the lungs, carbon dioxide pressure is lower to only 40. The bicarbonate in plasma enters red blood cells again. Now, carbonic anhydrase reverses the reaction. Bicarbonate converts back to carbonic acid, which converts back to carbon dioxide and water. The carbon dioxide is released from red blood cells into the alveoli. Then it is breathed out. So, bicarbonate carries carbon dioxide from tissues to lungs and then releases it for exhalation. So, to summarize carbon dioxide transport, 70% is transported as bicarbonate ions. This is the major route. 20 to 25% is carbaminohemoglobin and 7% dissolved directly in plasma. Every 100 milliliters of deoxygenated blood delivers approximately 4 milliliters of carbon dioxide to the alveoli. So we have three different methods working together to remove this dangerous metabolic waste efficiently. Your breathing is not random, no. It is controlled by your brain. Specifically, there is a special center in the medulla region of the brain called the Respiratory Rhythm Center. This center is primarily responsible for maintaining the breathing pattern. It generates the basic rhythm of breathing, the regular in and out pattern. This center has an intrinsic rhythm, meaning even if it was isolated from the rest of the brain, it would still generate rhythmic breathing signals. But that is not the only center. In the pons, another region of the brain, there is another center called pneumotaxic center. This center does not generate the rhythm. Instead, it moderates, fine-tunes what the rhythm center is doing. How? By reducing the duration of inspiration. It cuts short the inspiration so you exhale sooner. This allows the respiratory rate to be adjusted. Instead of always breathing at a fixed rate, you can breathe faster or slower depending on what the body needs. The pneumotaxic center prevents lungs from overinflating. But neural control alone is not enough. The body also has chemical control. There is a special area called chemosensitive area located right next to the respiratory rhythm center. This area is highly sensitive to two things, carbon dioxide and hydrogen ions. When carbon dioxide levels increase or hydrogen ion concentration increases, meaning blood becomes more acidic, this chemosensitive area is activated. It immediately signals the respiratory rhythm center. When this chemosensitive area signals the rhythm center, the signal is to breathe more. Breathe faster, breathe deeper. This rapid, deep breathing removes excess carbon dioxide from the body. Gradually, carbon dioxide levels decrease and pH normalizes. And that negative feedback, once carbon dioxide is back to normal, the chemosensitive area becomes inactive. Very automatic system. Additionally, there are receptors in the blood vessels, in the aortic arch and carotid artery. These receptors also sense changes in carbon dioxide and hydrogen ions. They also send signals to the respiratory rhythm center. This is a secondary system providing backup. If something happens to the main chemosensitive area, these peripheral receptors can still detect problems and signal breathing adjustments. Now here is something interesting. You might think oxygen is important in regulating breathing, but actually its role is quite insignificant. Only when oxygen becomes very, very low, critically low, does it trigger respiratory changes. The main regulators are carbon dioxide and hydrogen ions. Carbon dioxide is the primary signal. If CO2 increases, breathing increases. It is that simple and elegant. Oxygen is more of a backup system for emergencies. Asthma is a respiratory disease many people suffer from. What happens in asthma? The bronchi and bronchioles, the tubes carrying air, become inflamed. Inflammation means they swell up. When they swell, the diameter of the tubes decreases, they become narrower. This makes it difficult for air to pass through. You have difficulty breathing. And as air tries to squeeze through the narrow passages, it makes a sound. We call this wheezing. That characteristic whistling or squeaky sound. What causes this inflammation? Often it is an allergic reaction. Some people's immune systems overreact to certain substances in the environment, dust, pollen, pollution, certain foods. This overreaction causes inflammation in the airways. Sometimes stress or exercise can trigger asthma attacks too. The exact trigger varies from person to person. One person might react to dust, another to pollen. Emphysema is a serious chronic lung disease. In emphysema, the walls of the alveoli, those tiny air sacs, get damaged. They are not just inflamed like in asthma. They actually break down. The walls deteriorate and adjacent alveoli merge together, forming larger but fewer air sacs. This means the total surface area available for gas exchange decreases dramatically. Oxygen cannot be picked up efficiently. This damage is usually irreversible. Once done, it cannot be repaired. NEET emphysema is cigarette smoking. The chemicals in cigarette smoke directly damage the alveolar walls. This damage accumulates over years. One cigarette might not do much, but decades of smoking causes progressive deterioration. Other causes can include air pollution and occupational exposure to harmful substances. But smoking is by far the biggest cause, and the best prevention is to never start smoking or to quit if you already smoke. Occupational respiratory disorders are diseases that develop because of exposure to harmful substances at work. Certain industries produce lots of dust, for example, in grinding industries, where they grind materials. In stone breaking industries, where stone...
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.